My name is Dr. Patrick Soon-Shiong, and I'm here today to present to you the science behind the coronavirus. I really think it's really important for us to present some of the science because it's very clear to me that there's a lot of fear out there, and what adds to the fear is confusion. But if we could present the science and really try and understand not only how does this coronavirus work, how does it infect the body, what does it do, and how we can actually attack it, we have an opportunity to really provide some peace of mind, some level of control in the hands not of every individual, but the entire community. I'm really excited by the fact that this coronavirus has attacked us in 2003, and we have the benefit of that insight and scientific knowledge. But we also today have tools that we didn't have then. We have more genomics, we have more bioinformatics, we have supercomputing. And together with those tools, I think we have a real shot at not only defeating this virus, but overcoming this pandemic. So in order for me to take you through this complex story of this coronavirus today, we will talk about how do we understand this virus? How does the virus infect your body? And more importantly, explain the symptoms that are associated with this infection. What can we do to control and prevent its spread? And excitingly, what is on the horizon that the entire world scientists as a community are working together to cure this infection and preventing us from getting this in the first place? There are some questions we won't be able to fully answer yet, is how long is this pandemic, will this pandemic last? I think it's important that this 1 hour special will have a separate, maybe a slightly different lens. While the world is working on very significantly on how to kill the virus, we want to take this lens of what does this virus do to your body, i.e. the host, and how can we prevent it getting into your body, into the cells? How can we actually treat the host or the patient to prevent it becoming a severe fatal disease? So while on the one hand, we were working hard to develop a vaccine, and I think it's important and realistically important for us to identify a treatment now for those that are severely ill. Let me start on what is the coronavirus. You've seen a lot of names: coronavirus, COVID-19, SARS-CoV-2, SARS, and they all in essence mean the exact same thing here. This virus is a RNA virus. What I mean by that, the genetic signature inside this virus, that little curly, is made of a RNA that can reproduce. But most importantly, the virus has on its surface an envelope or a capsule. And then you see the spikes. And the spikes form a crown, and that's why it's called coronavirus. In 2019, it created this disease in China, and that's what COVID-19 was the first name. Coronavirus disease in 2019, COVID-19. And now the World Health Organization has created an official name. This is the latest scientific name, SARS-CoV-2. And why SARS-CoV-2? Because this is a second generation daughter virus of SARS. And why is that important to us? Well, it's given us a head start. It's given the scientists a head start since 2003, 2004, 2005 to fully understand this family of coronaviruses, specifically SARS and now SARS-CoV-2. What's exciting about that is we now have in our fingertips supercomputing, genomics, bioinformatics that wasn't as advanced even in 2003. And this is now allowing us to understand how contagious is this coronavirus. Because sadly, this mutation has allowed SARS-CoV-2 to become 1,000 times more infective than that of SARS. And that is the fear. The fear is SARS was an endemic, limited in its scale and scope, but now SARS-CoV-2 is a pandemic. So the question that we need to understand is, how does this enter your body? What does this do? How does it infect the cell? How does it affect the lung? And what organs does it affect? I think this is an opportunity for us to take a different lens of understanding the host effects rather than just the virus itself. And if we look at that host effects, we now truly have a shot at trying to block it and block the ability to get to SARS. So firstly is an infected person has the ability to contaminate 2.6 other patients. And how does that happen? Well, it's droplets, and this nanoparticle of a virus forms droplets, and these droplets can go on surfaces, and they go into your hand. The important fact is that your hand is now a vector. We'll come back to how we can use that fact to our advantage and kill the virus. But the lungs, what happens inside the cell? Well, this virus has now hijacked this receptor in our body called the ACE2 receptor. This receptor is on blood vessels, this receptor is on kidney, and even on heart, but most importantly on what we call the alveolar cells of the lung. It's in a very important receptor called ACE2 because it's there in your body to protect your lung tissue from injury. What has happened is this virus, through its spikes, has figured out a way to interact with that receptor and use that receptor to dock. The virus goes on now to take advantage of the cell itself and use the cell as a factory. It's now taken on what we call taking the RNA inside the virus itself, it breaks it up and remarkably takes what we call the Golgi apparatus, the machinery of the human cell to reproduce itself and repackage, and in so doing, it propagates. So this is the issue of how the virus actually regenerates and sheds. Now we are full circle because that shedding of that positively charged patient has the ability to contaminate others. And the opportunity, however, to stop, not just unfortunately the shedding, but stop that propagation is what containment is all about.